Hello, my name is Pat Eisenman. I'm a professor emeritus at the College of Health, the University of Utah, where I was on faculty for 34 years. I'm very pleased to be here today with you, and we're going to be talking about training the proper energy systems. What we're going to approach that topic through are these 4 points. We're going to look first at training energy systems and why that's important for you as a strength and conditioning coach. Then we're going to talk about what the systems are and briefly how they work, and then how you can target them for training. And then last of all, we'll consider, well, how trainable are they really? Okay, with that in mind, let's start in with looking at the importance of being able to select and train the appropriate energy systems. As you all know, the primary goal of program design, which you're involved in every day, is to make sure that you're helping to reduce the risk of injury, and you're trying to improve the performance of your athletes. And those are really closely tied, because an athlete who is fatigued is also more likely to be injured. So with that in mind, let's focus mainly now on the concept of improving performance by training appropriate energy systems. There are many aspects, and I'm not saying that any of those are less important than energy systems. I'm just saying that because of time, we are limited to considering only one of these many factors that influences performance, and that's energy availability. So right now, perhaps you're scratching your head thinking about, well, what in the world do we mean by energy availability? Well, for our purposes today, energy availability is having enough ATP available fast enough and long enough to reach peak performance. So the key in there is ATP. And ATP is a substance made of adenosine, ATP is a substance made of adenosine, ribosome, which is a sugar, and 3 phosphate molecules. The importance of it is that those phosphates are attached to one another by bonds, and those bonds contain energy. So in other words, left to its own devices, the phosphate would separate. But because the bond is there, it has a lot of energy to hold it on. Think about when you were a kid and you were playing crack the whip. The person at the end of that whip had to hold really tight or they would fly off. That's what the bonds do. They keep in this case, the phosphates together. That energy is the only energy in that bond that can be used for muscles to contract, nerves to send signals, and the other machinery in the cells, producing proteins, etc. If you don't have the energy from that specific bond, nothing works. And in fact, that's what death is, the absence of ATP and the breaking of that bond. That process of breaking that bond, as you see here on the right side of the cartoon, is releasing a phosphate. But the most important part of this is that it's useful energy. I also would like to draw your attention to the right side of the slide, where it talks about why we consider this to be immediate energy. It's only one chemical reaction, that's quick. The ATP is on site, it's in the muscle, you don't have to transport it in. And it's triggered by the nerve signal, the same signal that initiates the muscle contraction. Therefore, we call this release of energy from that phosphate bond immediate or explosive energy. Over here on the right side, as you look at it, the curve rises very sharply. That's increase in peak power is achieved very quickly because of the immediacy of this energy availability. And we've kind of likened it to an explosion. So explosive energy is really coming from the release of that ATP. This ATP is involved in all cells, all muscle cells. We're mainly talking about skeletal muscle, but nonetheless, ATP is found for all of them. And the key point here we don't have an unlimited supply of ATP in individual cells. It's very finite, which means that we have to get some biological energy so that we can remake or resynthesize the ATP. So I'd just like to draw your attention to the left side of the slide where you see the biological energy. That energy allows the phosphate, and the arrow is going toward the green arrow. You're going to put that phosphate on and convert the ADP at the bottom of the slide to the ATP that's at the top part of the slide. So that's critical if we're going to have continued muscle contraction or anything else. We have to resynthesize or remake the ATP. So looking at that again, biological energy, and I've said biological energy, but we're going to look at what that is here in a minute. But really the key to thinking about targeting energy systems is that we're trying to target a system that will replenish ATP at a rate that helps facilitate the performance that we're trying to achieve. So we can now address that question of where that biological energy is coming from. We're going to look at three systems, each of which provides the energy to resynthesize or remake ATP. One of them is the phosphagen system, the fast glycolytic system, and the slow glycolytic system. We're going to just start here at the first one, the phosphagen system. And by phosphagen system, we're talking about phosphocreatine, which you see over on the left side of your slide. So we have the phosphate group and a bond, which I circled in green, attached to the creatine. All right. If we break that bond, then we provide the energy to resynthesize ATP. So in the bottom part of the slide there, you see the phosphate now plus a creatine plus energy. So they're separated. The bond has been broken. Now the energy can move over and allow for the replacement of the phosphate and the energy to resynthesize or to drive ADP to ATP. That system is called the phosphagen system because it's so focused on the phosphates. In this slide, we see that when you're doing a 1 RM back squat and then you move to a 3 RM, most of the energy, the primary or the prominent energy system that's being used to resynthesize ATP is the phosphagen system, or the breaking of creatine phosphate. And that's represented here in the red part of the bar graph. Now it's not 100%, but most of the ATP is coming from that phosphagen system. And that's what allows the incredible power to be demonstrated in the back squat, and then moving a little less power as you're going to a 3 RM. And again, the same slide that or the same drawing that we saw earlier, where you have a very rapid rise in the power that's being produced by the muscles because of the quickness with which phosphocreatine can break apart, provide the energy to resynthesize ATP. But again, we have this caveat. We have a finite amount of phosphocreatine. It can't go on forever. So as you look at the graph, the line graph, you see that the power is falling off. It's getting lower and lower over time. That decrease is because we've had to shift to another energy system, and that's the fast glycolytic system, which we're going to consider here next. Before we actually look at fast glycolytic systems, I'd like you to take a look at these numbers presented in this chart. They're ratios, which means that there are no units. Okay, so as to take a closer look at those numbers, first of all, phosphocreatine is stored and it rapidly replenishes, as we've just said. And look at how much more, relatively more phosphocreatine is in the muscle than ATP, almost 5 times as much. Now, at fatigue, look at how little phosphocreatine is left in the muscle. But look at ATP. It's dropped very, very marginally. So that is because the system is set up not to allow ATP to go down, and conversely, not to allow ADP to increase much. In fact, the rigor mortis that sets in with death is because ATP is no longer being resynthesized. And so muscles can't contract and they're in rigor. The other point down at the opposite is with creatine. Very little free creatine is in the muscle, but at fatigue, there's a lot more because the phosphocreatine has been split apart and energy used to keep this ATP from falling down. So not much ATP concentration decrease is allowed. On the other hand, a fair amount of phosphocreatine decreases from the muscle, and it's accompanied by exhaustion or fatigue. So we've looked at this first system that helps to replenish ATP, and that's the phosphagen system. And we've used the analogy of an explosion to talk about how quickly this system can replace. But the limit is it can't do it for a very long period of time. So we have to have another system that can take over at a slower rate, but still nonetheless replenish the ATP. And that system is known as the fast glycolytic system, or perhaps you've heard it called the lactate system. And I'm going to use the analogy here of a car, a race car, high use of fuel, a lot of power, but not nearly as much power as you would have in an explosion. So at this point, you're probably saying, what in the world is the fast glycolytic system? We're going to not go into it in a great deal of depth. I just want to highlight a few points. Glycolysis breaks apart glucose. So we have a glucose molecule here, and it's got 6 carbons. That's what the C stands for. And the black lines are the bonds. So this process of taking glucose and breaking it down to pyruvate is releasing the energy from those bonds. And that energy, as you see down here, is used to resynthesize some ATP. Now, what happens to pyruvate depends upon the circumstances in the cell. And when we're talking about fast glycolysis, we're talking about the production of lactate. So it can occur very quickly. Pyruvate is converted to lactate. There isn't any direct manufacturing of ATP here, but it allows the circle to continue. It allows glucose to be broken down and some energy to be produced. So that diversion over to lactate is important because it allows glucose to sustain its activity. So fast glycolysis is when there's pretty rapid synthesis of ADP. So you're working hard, and you can quickly convert pyruvate to lactate. And I've used the term down here, pretty rapid, because it's not as rapid as phosphocreatine is able to replace ADP to ATP. So it's a little slower. But it does allow the process to continue and allows some ADP to be changed to ADP. So this concept of pretty rapid is again illustrated here in some diagrams. Power increases pretty quickly. Velocity is high, and then it decreases as you go further distances. So the initial peak power and the faster velocities at shorter distances are supplied by energy from the phosphagen system. As the duration of the activity increases, there is more contribution of fast glycolysis in the resynthesizing of ATP. So you also saw now that we had a number of reactions in the fast glycolysis pathway. That makes it go slower than the phosphagen system. We're going to talk more a little later, but for right now, it's critical that you use glycogen and not necessarily just blood glucose for this to go as quickly as it can. And the third point is that it's again tied to a need. So as soon as you start releasing hydrogen ions from the breakdown of ATP, you stimulate fast glycolysis. And the fast glycolysis provides the energy to resynthesize ATP from ADP. So again, let's go back to the drawing that we had earlier. And now we're adding some little bit longer distances. So therefore we have more time in which the muscles are contracting. And you notice that the contribution, the relative contribution of the phosphagen system is coming down. You still have to have energy, but you can't provide it as faster rate. And so the speed or the velocity is decreasing as you go to these longer distances. Now you notice that we have some blank spaces over here on the graph, and that's going to lead us into the third of the energy systems that we're going to talk about. And so that third system is slow glycolytic processes, or the slow glycolytic system. And the drawing that I'm going to use here is the compact car. It sips fuel, goes a long way, but not necessarily quickly or powerfully. And so that's the characteristic that we're going to talk about here as we talk about slow glycolysis. We're also going to point out this notion of glycogen. So as we looked at glycolysis before, we use glucose up here, which is a 6-carbon chain. And we went through all these chemical reactions to get down to pyruvate. What's really happening is we're splitting that pyruvate into 2 3-carbon chains, and we're producing lactate. But we still have a lot of energy in all of these bonds, in these 2 molecules of pyruvate. If we can direct them here to the slow glycolysis or oxidative system, we can capture more energy. It's just going to take some time. And again, since we've talked about chemical reactions taking time, as you look at the Krebs cycle here, you see that there are a lot of chemical reactions taking place, which take time. So we have our pyruvate, this 3-carbon chain, half of the glucose. We're going to, again, break one of the bonds, convert pyruvate to acetyl-CoA, which is a 2-carbon chain. And then we're also going to have that 2-carbon chain join with a 4-carbon to form 6-carbon citrate. So we have 6 carbons here at the beginning of the process. It's going to go all the way around here, end up at 4 carbons. And in the process, hydrogens have come off from breaking of bonds. Carbon has come off from breaking of bonds, releasing energy so that we can resynthesize ATP from ADP. So again, to highlight these, you're going to break bonds to release energy. That energy is going to be used to resynthesize ATP from ADP. So here's the energy that's being used to move the equation in this direction. We're going to use the hydrogens and the carbons along with oxygen to produce water and carbon dioxide and the energy. We're not producing, we're just transferring it to ADP so that we can remake ATP. It takes time and it takes oxygen. That's part of the reason it takes time, because you have to transport oxygen in. And the energy released from slow glycolysis is also used, a lot of people don't think about this, is used to replenish the phosphocreatine stores. Now, another aspect of why this takes time, all of these chemical reactions are taking place in the mitochondria, which is an organelle inside of the muscle cell, and other cells too. But you have to transport, there has to be a protein that takes pyruvate in here and carries also some of these byproducts out. All of those transport functions take some time. But then let's plug in the last 2 bar graphs here, and we see that as you extend the duration of the activity out here to 1,600 meters in a marathon, you have a decreasing contribution of phosphagen. That doesn't mean you aren't using it. It's just not the dominant system. And the phosphagens are being replaced continuously, and they're being replaced with slow glycolytic processes. So this gray bar represents the fast glycolysis, and The black represents the slow contribution, the contribution of that system to energy. You might say, well, wow, why is there still some fast glycolytic in a marathon? How can that be? Well, as you train, slow muscles use lactate as a fuel. So the idea that lactate is a waste product is not true at all. But you have to train specifically to get that. And we're not going to talk much about that kind of training because the marathon distance is not used in college or by collegiate athletes. So the take-home message is from taking a step back and looking at these three systems is that they, although there are three of them, they do not operate in isolation, but they do limit performance. Now, you've probably all seen charts like this relative intensity chart, and you're well aware of the fact that as the number of repetitions increases, the intensity of the effort is going to decrease, then you can choose to have it even lower by making conscious determinations. So it's not a new concept for you, but what we've done that may be new for you is link the concept of energy systems to this notion of these intensity charts. Both ideas, the intensity chart and the time that's involved, and the energy system function are key to the rest of this presentation. So hopefully we have made the case that energy availability is fundamental to performance. And so you're probably saying, well, how can we increase this functioning of these systems so that we can better allow our athletes to improve their performances? Well, there's two ways that we could train systems. One is that if the energy substrate, the stuff that's being used to break bonds and release energy, if you could increase that amount that's stored in the muscle, that would work. So if you could increase ATP storage, that would work. Or if you upregulate, or in other words, increase the speed with which the processes in slow glycolysis take place, that would also help. So let's look then at these systems and how we might target the system and identify them so that we can help athletes maybe improve their performance. The topic of identifying the proper energy system, there are really a couple ways you can approach this. One, you can consult the literature. You can look at, there are myriad studies that have been done and lots of data that's been published to show the relative contributions. Here is a table that talks about ranking the importance of the systems. And a 1 means that it's not very important. So in the 5,000 meter run, the ATP phosphocreatine system is not particularly important. However, using muscle glycogen is. And then the other kind of notation here is to look at the 400 meter swim. Look how these numbers look very similar to the 1,500 meter run. Can you think of why that might be? Well, the amount of time that it takes a good swimmer to go 400 meters is about the same amount of time that it takes the 1,500 meter to cover that distance, assuming that they're both well trained. So in other words, we see this time factor being central to the contribution of the energy system. So you can find other categorization schemes that take sports more so than runs like we see and swims like we see over here, and talk about the kinds of efforts that are involved in those sports. And you can use that information to help you think about which systems, energy systems, are going to be fundamental or going to be used. But I would like to suggest that you can also use your knowledge and your creativity. There was a kind of a cool article published by Jeffries a few years ago, and he identified 5 minds that are characteristic of the modern strength and conditioning coach. The first one is that you have the discipline to study what's going on, that you take this information that may be kind of theoretical and you synthesize it and put it together into some ways you can use it practically, and that you do this synthesizing in a very creative way. So I am suggesting that your use of the clipboard to observe what's going on in the individual athletes that you're responsible for coaching. You can use things as simple as a stopwatch to determine how long the activity takes, or a measuring stick, how long they go or how far they go. And you can go all the way up to some really sophisticated, and this is just an example of a global positioning satellite system. So the satellite bounces information from here up to the satellite and then back down. This happens to be soccer. And so they are looking at, each player has one of these on, so they identify how far they run, how fast they run. It's all computerized, and it's pretty sophisticated, but you can do many of those same things with a stopwatch and a recording system. And the fact that you're asking questions about what you should be measuring is the way you use your creativity. And again, you see that notion here of duration, short periods of time, high intensity, very reliant upon the phosphagen system. As the duration of the activity gets longer, you're more and more reliant upon slow glycolysis or the oxidative system. So this is moving up and down. I'm suggesting that you use your knowledge of the athletes that you're working with. How many times do they have to run down the field? How much relief do they get? What style do you have in football? Do you have time between each set, or are you having a hurry up offense? Those kinds of things influence which system is also being used. So regardless of whether you're going to use a global satellite positioning system or a stopwatch, it doesn't matter. The creativity of you and your knowledge as a strength and conditioning coach is what's critical. What are the questions that you are posing? Whether it's how much distance is being covered, how many times, how much time between executions, all of those things. We're going to determine which energy substrate is being used. And then you're going to want to know, well, can we increase that substrate in the cell? Or can we increase the resynthesis of ATP? Those questions will, those statements will flow out of your answers to these questions. So the stopwatch is your friend. It tells you how long it takes to execute. But remember, the intensity has to be there too. When you start walking across the room, you're using the phosphagen system for the first couple of steps. But then, because you're only walking, that system becomes very easily replenished. And so you're really then dependent upon the slow glycolysis system. If you jog across the room, you use phosphagen again, you drive it down a little lower, but still, you're pretty easily replenishing the ATP with the slow glycolysis system. As the intensity of the activity increases, the time that you can maintain that intensity decreases. And so that relationship is fundamental to thinking about it. And it's also fundamental to us thinking about how trainable these systems are. Unfortunately, there isn't any clear evidence that supports the fact that training can increase the amount of ATP or phosphocreatine that's stored in the muscle. That's kind of a bummer, huh? But, and it is a pretty good size, but resistive exercise can increase the absolute amount of stores by increasing muscle mass. So here you see a marathoner who has relatively small muscle mass, and so therefore absolute quantity of ATP phosphocreatine is not large, but it's not a major source of prominent energy system in running that 24 miles. Whereas the sprinter, this world-class sprinter, has lots of muscle mass, therefore lots of stored ATP phosphocreatine, which supports the power that he's able to achieve in that 100 meters. The next thing that exercise and training can do is increase and tweak the neuromuscular activation. So activation of fast twitch fibers in particular that are large and therefore have a fair amount of ATP phosphocreatine in them to begin with, they are going to be able to be called upon more by the trained person. The untrained person can't activate all their fibers. The better trained individual is more able to come closer to activating all of them. And then last, the amount of recovery time between exercises or exercise bouts allows for you to target. So if you're trying to train the ATP phosphocreatine system because you've identified that that's important for your explosive athletes, then you want to be able to make sure that that system is tweaked, it's stressed. So that's why you have the longer work rest ratios. 1:20 is probably a starting point for 100-meter sprints. 1:2:10 for high intensity, more lower volume. So if you look over here at the curve, this is the recovery of phosphocreatine. So in other words, how much time does it take phosphocreatine once it's been driven down? So this is the exhaustion, the fatigue. It's a very low amount of phosphocreatine in the cell. How much time does it take it to get up here to this plateau where it's essentially replaced? So you're looking down here and you see it's like 2 to 3 minutes. And that's why you have these long recovery periods, so that when you start the next exercise or the next bout of exercise, you're going to be using the ATP phosphocreatine system. If you diminish that recovery period, when you start the second bout or the third or the fourth, you're going to be using fast glycolysis. You're not going to be stressing and using the phosphocreatine system. So it's interesting that in a lot of plans for example, this daily training session, it's not until you get down here at the bottom that they talk about sport-specific energy system training. I would like to consider that you can also use it throughout the session. So if you really are trying to develop speed, which is dependent upon the phosphagen system, you're only going to use that system if you allow ample recovery time between exercises. The same thing is true for jumps and throws and the big compound movements. Adequate recovery means that you are going to be stressing the system that you're trying to stress. You're going to increase the activation of fast twitch fibers. If you condense the recovery period, you're using fast glycolysis, not the phosphagen system, because the speed, the performance goes down because you simply cannot sustain it, because you haven't been able to recover the stores of phosphocreatine. Okay, so let's look then, since we've mentioned it several times, the fast glycolytic system and see how trainable it is. All right, we're going to now talk a little bit about the difference between glucose and glycogen here. And we are talking about this system as being pretty high power, moderate capacity. And if you use glucose, as we see up here, you have this series of reactions. However, if you use Okay, we're now going to talk about fast glycolysis. Remember, that was the system that took glucose, went through a few chemical reactions, and had produced enough energy to resynthesize a small amount of ATP pretty quickly. And so that's why we have this notion of pretty high power and moderate capacity. It's true that you may be able to increase muscle glycogen, which would be helpful because that increases the substrate that you need for fast glycolysis. And the reason the word may is there and why this matters is because of the relationship between muscle glycogen and blood glucose. Glycogen is a huge molecule with lots of glycogen or glucose molecules stuck together. Core protein, branches of glucose units. So each of these is a glucose unit, and I've just highlighted one of them. When we break that off, that enters the pathway, the glycolysis pathway down here. So as you notice, that allows us to bypass that splitting of ATP, which means that we're saving energy if we can use stored muscle glycogen rather than blood glucose. It's faster. Glycogen is stored on site, and it helps to keep the hydrogen ion concentration down. So to recap, fast glycolysis really for athletes, explosive athletes, is coming from stored glycogen, not from blood glucose. It saves ATP, slows the production of hydrogen ions, and allows the system to run a little bit longer. Okay, and that's where you see the saving of ATP, and you see this hydrogen ion. It's not going to be broken off, and that's what this means by slowing the production of hydrogen ions. So training may increase muscle glycogen a little bit. Probably more importantly is diet, making sure that you have adequate carbohydrates that you're consuming. Let's move then to the second points in terms of training the activity of the glycolytic system. Those are many enzymes that allow those reactions to take place. With training, those enzymes work better. And so the goal then, if you're trying to train the glycolytic system, is to keep the rest interval short enough to stress fast glycolysis. In other words, you don't want enough time to replenish the ATP store or the phosphocreatine store. So this is really kind of the opposite of what we were talking about when we were talking about training the phosphagen system. So the duration of the rest interval depends upon the needs analysis. In other words, how long do they have in the competitive situation? And it also depends on the annual training plan. While you might be trying to train fast glycolysis, you don't want to do it too early in the annual plan because it can lead to overtraining. So it usually occurs later in the preseason, and then a little bit you maintain it in the in season. So again, it boils down to, what does your analysis say? How long do the kids have to recover in the competitive environment? How long do they have to go? How many times do they have to do that? That helps you set the number of repetitions that you're going to use in your training. So those questions and the creativity that you use in asking them and answering them is really key to being a great strength and conditioning coach. Okay, let's look now at the third system, the slow glycolysis system. And remember, here we have the pyruvate, which is 3 carbons, goes into the mitochondria, converted to a 2 carbon, which means we're going to release some energy. And then we can also have fats through beta oxidation enter this, and you chunk off the last 2 carbons, and it goes into this cycle. It takes time for this to take place. You have to transport in, you have to have the substrate, you have to have fatty acids or pyruvate being brought in, you have to have the delivery of oxygen, you have to have the removal of CO2. So it's a slower system, and thus the term slow glycolysis. It, however, is a very trainable system. It has low power, but it has a really high capacity. And it's resynthesizing both ATP and phosphocreatine. So even the power athlete who has to do the activity, whatever it is, the run, the jump, the block in volleyball, multiple times, needs to be able to rapidly resynthesize ATP and phosphocreatine. So there is a need to train the slow glycolysis system, but just maybe not in the same way that the endurance athlete would train it. So unfortunately, when we talk about the aerobic system, we all jump to thinking about marathoners. And I just want you to keep in mind that they have a totally different demand on their system than the power athlete does. So the question is, can we train the slow glycolytic system and not lose the explosiveness of the power athlete? And the answer to that is yes, we can. It's not by using long distances at slower efforts. That does do some good things for the oxidative system, but the risk is that it detracts from the quality of the anaerobic training, of the power training. However, the use of high intensity interval training, HIIT training, allows for the slow glycolytic system to also be used. And you say, well, how can that be? Well, remember, the faster that system can work, the better you can replace the phosphagen system. And therefore, the power athlete is going to benefit from it. The risk is if you start it or do too much of it, you're going to overtrain the athlete. So to recap, that trainability of that oxidative system is excellent. You can upregulate the enzymes. You can increase the size and the density of the mitochondria. Remember, that's where the action is taking place. You can increase the ability of the heart to move oxygen delivery more efficiently. You can increase the delivery of blood and the removal of CO2. And you can get lactate around to cells that can remove it. And you have faster recovery of the PC stores. So let's take a look at where we've been today. We've looked at the energy systems and the importance that they play for performance. And we've highlighted the fact that performance is really one of your key goals as a strength and conditioning coach. We've then taken some time to look at the energy systems and how they work. We identified 3 systems: the phosphagen or ATP phosphocreatine system. We have identified fast glycolysis and slow glycolysis, sometimes called the aerobic system. But we have also noted that they work together. You're not just using one of them. One of them dominates in certain kinds of activities, but you're not just using one of them. The third point is that we have spent some time thinking about, and I've been challenging you to use your creativity in thinking about how you might target those energy systems. And remember, the key to targeting the appropriate systems is to think about what's required, what's the needs analysis on the field, on the court, on the track, whatever type, in the pool, whatever type of athlete you're working with. And then how are you going to measure that in terms of, and that will help you structure how long your time periods are, how much relief you give people between sessions of exercise. And again, I come back to this notion of creativity, spending time thinking about it, spending time talking to the coaches about, if it's a team sport, what their philosophy is, what are they trying to push the ball up the court in basketball, or are they taking more time, or are they more deliberate? Those sorts of things, and how you creatively assess that, are going to dictate how you structure, how you design the programs that you're using. One size will not fit all. And then we spent a little bit of time talking about, even if you do a great job, are all of these systems equally trainable? The truth is that the phosphagen system, in terms of increasing the stores of ATP phosphocreatine, probably is not great. But resistance training increases the absolute size of the muscle, and therefore there's more stored. The glycolytic system is quite trainable. The trick is to not overtrain the athlete by pushing too hard. And then the aerobic system is very trainable also. The power athlete will probably want to be using HIIT types of intervals so that you don't interfere with the quality of the adaptations that take place with your anaerobic training. But it is very trainable, the aerobic system. So my message would be to consider those HIIT training sessions, anywhere from 10 to 30 seconds. with 4 minutes to 2 minutes, as you progress in the intensity of the training for those anaerobic athletes. So we've looked at these 4 questions, considered them in light of how they might interact with the performance of the athletes that you work with. Now your challenge is to think as creatively as you can about how you can use time and intensity assessment to identify what your athlete needs, and then try and match that with the kind of training regimes that you put together. Hopefully this has been helpful for you. I've enjoyed my time with you. Wish we had been here in person. Thank you.